Invite other friends to join. Oh, okay.
Hi, how are you? Sorry,
Hi.
putting up the topics. I hope if I don't talk much for a few minutes, it's because I'm putting up the slides and everything. But welcome and thank you for coming. Definitely.
Thank you.
We are talking to each other through Zoom, which is why there's echoing before we sent you the slides. So I think we ought to probably log out.
We'll be back, Karina.
Yeah, it's not too bad. I mean, yeah, it's not horrible, so it's good.
And we sent the slide set. Did you get that? We
Yeah.
realized that we didn't put the numbers in there, so I sent you one with the numbers. Oh, perfect.
Yeah, if you can, please check if you can access it and.
Okay.
Yeah. But okay, that's great. And now let me add in the chat. I usually share lab websites so people can, check out more.
I think this one is the one that doesn't have the slide numbers on it. I think I sent you a second email, but it was about 10 minutes ago. Is it too late to update the
Oh, no,
PDF?
it's not.
Okay.
Oh, you did. Oh, I'm so sorry.
Yeah. I'm so sorry. It should say edited with it.
Okay, no problem. I will.
Thank you.
It just takes a couple of minutes, but we have that's why we
Yeah.
do that. That's why
Yeah, thank you.
and yeah. Yeah, it's uploading, so we'll be there in a couple of minutes.
Okay, great. Thank you.
Hi, quick question. Maybe you could incorporate that in. When you say Africans, are you referring to West and Southern Africa, or that include North African? Because North African history actually in America is very long. Napoleon brought many from Egypt to Morocco to fight with him in Louisiana, and so is the Spanish-American Warfare.
Mm-hmm.
So, and there were many, but those are fair skin. Most of them are fair skin, blue eyes, Mediterranean.
Yeah. Most of our results are indicating West and West Central Africa along the African coast. Yeah, North Africa has an extremely complex and rich history, but for this study, we're focusing mostly on along the West African coast.
No, that's good to know. I was just trying because the word African is a bit too broad.
Yes.
Yeah, I'm sorry. We have very limited character number for the title, and it's always really tricky to make a title that is accurate enough, but then still fits into the limitation. So,
No, perfectly fine. I was just curious because that's an interesting topic. That's all.
Thank you.
yeah, thank you. And we will start in a couple of minutes. So yeah, stay around to ask more questions. Always more interesting when people participate.
Yes.
Okay, I shared the lab websites. The new file should be up.
Yes.
And yeah, now I'll share on Twitter that we're really starting now. And
Tad, do you know you're on mute just in case?
So this is not the right slide set. I made modifications to the titles and colors and things of this. This does not have numbers on it either.
The edited version?
This does not have this is not the version that I sent back to you, Raquel, that we've worked with. It has all number of things that are in different colors and other things that we'd worked on.
Okay, 1 second, Karina. I might have accidentally saved the wrong slide set. Let me check.
oh yeah, don't worry. We still have a few minutes. I mean, it's more. No, I didn't. I'm not in a time. Like I'm not in a rush, so it's fine.
Yes, that was my fat. Katarina, let me send you the updated. I had two versions of this open and then I edited the wrong one. So let me quickly just do this really fast.
Yep, no worries. And thank you, Theodore, for noticing it.
Yeah.
Well, we both spent a lot of time making these kinds of slides and going back and forth and editing them, so we're pretty familiar with the details. And
Yeah, well, thank you for taking the time to do this for this presentation. I know it's, you have a lot of other things to do. So yeah, I really appreciate it.
Okay, the updated one should now be sending to you.
I'll get it, and yeah, now there it is.
Yep.
Great.
Okay, that one has the. Thank you for catching that, Tad. I had two versions of the slide set open and I accidentally didn't even notice I was editing on the wrong one.
Yeah, and in the meantime, everyone in the audience, feel free to check out the lab website and also the paper. I shared it, and so, and also if you have questions, please, or comments that you want to get addressed, please either send them to me, put them up in the chat, or or just raise your hand and participate in the discussion. And we will start in a minute when the slides are uploaded. And it's an honor having you, Raquel, and Theodore here. Thank you so much for taking the time again. And for, I know it's a hustle, like to make that count and all of that. So what was, let me just see, what was edited? The title now, because now I have a bunch of.
I know, it should be under edited 2.
Okay, it didn't upload. I uploaded it maybe somewhere weird. Yeah, now I have it. And I will. Okay, now I can pin the link.
Thank you. And thanks so much for having us today. This is a really great app. It's really exciting.
Yeah, I hope you'll enjoy it. Okay, now we are all set, and I know people will continue coming in, but let's start with introductions and then we'll go from there. So welcome everyone to Science Society, and of course a special welcome to you, Raquel and Theodore. And before we start, let me give just a short introduction, and then we go continue the introduction with like a short interview before we then dive into your really interesting research. So Professor Theodore, he is a professor of anthropology in the Department of Anthropology, and he is also a consulting curator in the University of Pennsylvania Museum of Archaeology and Anthropology, and he's the director of the Laboratory of Molecular Anthropology at the University of Pennsylvania. And he has been researching the genetic prehistory of Asia and the Americans for 30 years. And his current projects also include studies of genetic diversity in the indigenous population of Canada, the United States, Mexico, and the Caribbean. And also his group also looks at population history of Georgia, Pakistan, Kazakhstan, and others. He did his bachelor at the University of Georgia and his master's at Emory University, and then later also his PhD at the Emory University. And then Dr. Raquel Fleskes, she's at the National Science Foundation, a postdoctoral fellow at the University of Connecticut in the Department of Anthropology. I also shared her website in the chat. She is an anthropological geneticist. And her research falls at the intersection of ancient DNA, archaeology, and community engagement, which I think is really interesting, that you have this intersection. And to understand the population history of colonial period in North America. So yeah, welcome to both of you. It's really an honor. And we usually start by asking, so if you can look back at your life, if you remember, what drove you to become a scientist and to do this type of investigations? Was it maybe something in your childhood? Was it a childhood dream, or was something that sparked your interest later on? Thank you so much.
Who would you like to start first?
Both, like Theodore, since I introduced you first, just go ahead.
Okay. So I suppose I had a bit of a leg up on getting involved in scientific and work and biological research because my father was a biologist. And so I think I first got intrigued by science by reading his books and books on evolution and, you know, details of dinosaur history and phylogeny and so forth. So for me, that was probably, 8 or 9 years old. Became aware of this broader, diverse world, and also all the the wonderful illustrations in the textbooks of anatomy and diatoms and things of this kind, which were just stimulated the imagination for the way that trilobites, the way the world was created so long ago, and the way that it now looks today.
Yeah, thank you. That's interesting that your father was a great inspiration, and that is really nice to hear, because sometimes it can also do the opposite.
Yeah.
Like,
Mm-hmm.
but that's really nice to hear and encouraging for all of us that have children, that they won't be repelled but rather attracted to the subject. So that's wonderful to hear. Thank you. And Raquel, please go ahead.
I wanted to know. I wanted to be an anthropologist when I was 16 years old. I had read Anthropology for Dummies, which is the bright yellow book. And I didn't know how it was going to work. And my mother was very worried. She told me to major in business too. But I was really interested in thinking about how we can apply methods in genetics as another way of thinking about our past. And so when I joined to work with Dr. Theodore Schurr at the University of Pennsylvania for my PhD, he really encouraged me to think about learning how to do work in ancient DNA and then applying it to the study of the colonial past. So I grew up in Maryland and Virginia, and so I started out first doing projects in that local area, and that eventually led to work with the Anson Street African Burial Ground Project. And I think our background as anthropologists really allows us to integrate community perspectives, oral histories, archaeology, as well as the hard science, so to speak. And so we're really excited to be here today to kind of talk to you about this work that we've been doing. I think about 7 or 8 years now, Tad.
Yes. Yep.
Well, yeah, that's wonderful that you went ahead and went with your passion anyways. It's so interesting to me that I feel like, it's so culturally different than in Europe and many countries. Going into science is kind of prestigious and parents are very happy if you do that. But I heard from various colleagues that parents usually ask, "Oh, are you done with your science thingy and start making money?"
When are you
that's
out of school?
yeah, exactly. When are you going to the real world? It's really and thank you so much for staying in that field and doing that work. So, which gives us this opportunity to talk about this work. So, thank you. And then, if I don't know if there is maybe an interesting backstory how this specific project came about. Was it really hard also to get the collaboration? Was it easy also to get grants for this project? If you could share some insight with us, I think that would be really interesting. Thank you.
I think that those kinds of aspects of the project will come forward as we go through this slide set and explain how the project started and who's involved and what it took to carry out the work that we're going to describe here and are continuing to work through at the very moment.
Wonderful. So that's the perfect transition. Everyone, please access the slides through the link we pinned on top of the room. And yeah, the stage is yours. Thank you.
Okay, so Dr. Fleskes and I have divided this talk, at least in trying to cover some parts of it, but we may jump in and echo various points or add details to each other's comments along the way. This project for us, the project, I'm not going to. I'll get to a few points about its actual start, but she and I have been involved in this project now for a bit over 5 years, although the project was actually going on before that time. And so this represents what we're talking about now is an overview of that work and also reflections on how all of this got started in the first place. So in the second slide, you see the outline of this presentation that we're going to follow, including talking about its history, which we've just been referencing, who was involved with this, how what we do as part of this project, the ancient DNA work itself, which is at the central portion of the research that we're publishing, and Dr. Fleskes' lead. We'll talk a bit about some work that we have in motion now, which works with a different kind of DNA from dental calculus, and then talk about that where things are going at this point, and then make some acknowledgments, including funders, which you asked about. So this project, the current group that's here today that is working on all these details, the ASAGWA project, which stands for the African, the Anson Street African Burial Ground, the acronym, include the names you see here: Joanna Gilmore, Lachey O'Bray, Grant Mishu, Raquel, and myself. And we'll mention some other folks who've been involved along the way, including the person who led the project from its very inception, Dr. Adeo Funyan, in just a moment. But collectively, all of our team and others who have been involved in the different parts of the analysis, you'll see in the fourth slide, which is the central questions that we were asking, and also which the community was interested in asking or learning about, which is what are the contributions made by enslaved Africans and their descendants to the fabric of life in Charleston? How did they help Charleston actually be built and constructed, literally and figuratively? Since their participation in Charleston's history has been largely obscured in many ways, and which remains somewhat fragmentary, such that work like this becomes necessary to tell stories about the people of African descent who are brought to Charleston, and then whose descendants lived beyond that time. So the sixth slide shows Dr. Adeo Funyan, who was as you can see, he's you see the details about here. He began this work actually by um, mapping his own family graveyard and realizing that in fact many graveyards and family graveyards were not actually documented, and so he developed the Gullah Society in large part to try and do that work and map burials across all of the Charleston Peninsula and surrounding areas to help tell the story of African Americans to a greater extent. This involved a significant amount of engagement with community and at this time, and also working with others to help map graveyards, as well as creating this kind of a database that we were beginning to use for this work. But that was all in motion. That was started somewhere around, I think, 2015. But what really prompted Dr. Afunian to get us all involved in this project was the next slide, which is about the discovery of the Anson Street burial ground. This was discovered accidentally when the Gilliard Center in downtown Charleston was being expanded to encompass more space for public events and cultural events and so forth. And the human remains were found at that particular location, 36 individuals and 36 burials. And then they were removed at that time and put into safekeeping at a cultural resource management firm, and then later analyzed by osteologists and to begin trying to figure out who these persons were. And part of this story involves that answering that very question, who they are. So and then and I think it was in 2000 and maybe 17 that Dr. Ofunian and the Gullah Society were asked to become part of the process of reinterring these remains because they were determined likely to be of persons of African descent of putative enslaved status. So that got things rolling in terms of that plan for the reinterment, but also community conversations like we've been involved with at the next slide, both with before our involvement and also later when we became involved, which is to ask the community what they wanted to have done with the remains of the ancestors, as we call them, and then what kind of research into their ancestry and history and personhood that they wanted to engage with. And they were enthusiastic about using all available types of scientific means of assessing their history and ancestry. But each along the way, we had various conversations with them about what we wanted to do, what our results were. We work with community members to do their own DNA testing to find possible connections to the ancestors whose DNA we also studied, and Dr. Fleskes will detail in quite a bit length. And you can see then the next slide actually shows the way in which we engaged in this DNA work with both living and also the ancestors themselves and the people that we got involved with, that we involved in training, and including Adiemi Oduole, who you see on the far right of this slide under Raquel's tutelage. And so each step of the way, and much of the information that you see in the subsequent slides, we presented in person, as you can see Raquel and I, the DNA research updates slide, going through the details, both what we find for the ancestors and also for the persons whose DNA we tested in Charleston. We also, in the next slide, you see we elicited feedback at each step along the way about this process, what they learned, what they'd want to learn, directing us perhaps our attention to certain things they wanted further analyzed. And this was an enormously productive relationship and process for us to be able to guide this work going forward. The work we've done, especially based on the initial DNA evidence, but also other things learned about the ancestors, led also in preparation for the reburial a whole variety of artistic responses to the ancestors and to African American ancestry and heritage, which you can see then in the artist workshop and the community art exhibits in Charleston slides. Quite an extraordinary response, both at the sort of the adult level and then also at the level of middle school and high schools. And all this work you see represents that kind of response. The next one, where the tree actually I think also involves senior citizens actually using indigo batik to make this tree of life, in essence, that it represents African heritage and its deeper roots in Africa. The next slide, which is on let's see here, is about the memory of creating a memorial. And there's more that we can say about this, and maybe Raquel, I can talk about what was started, and maybe you can add towards the end what actually is in motion, if that's okay.
Yeah, that sounds great.
Okay, so the basic idea was that we were in the process of analyzing the remains and planning for the reburial and trying to figure out how best to honor them with a permanent memorial. So there were some initial steps done at the College of Charleston with students who were inspired by the work and who wanted to actually come up with an idea for recognizing the ancestors, these 36 persons who nothing much was known about before the discovery because there was no reckoning of their identity in any kind of historical records. And so this was the start of the process in 2018 for us to figure out how to actually build something that would acknowledge their personhood and their importance for Charleston. We also consulted with a well-known architect and designer about this and consulted about how to build this memorial, and we got feedback from community members about what that might look like. We also were able to do create based on the initial data we had, archaeological, historical, and genetic. We began to make profiles of all the ancestors based on the data that we have, and so the Restoring Personhood Changing Narrative slide shows at least a few of those kinds of poster-sized representations of persons, at least as we could best begin to summarize what we know in some sense at that point. So the key points, at least at this time, then, is that the project we made a significant effort to expand knowledge about African ancestors in Charleston, about whom not much was known. We began this memorialization process, and also plans to rebury. And also, we were able to do this kind of work, which had, this bioarchaeological work, which was socially engaged and designed specifically to counter sort of racist perspectives on African heritage and history, and actually give basically greater personhood and recognition of the ancestors whose remains that we are being permitted to analyze. So I'll turn things over to Dr. Fleskes now.
Thank you, Dr. Schurr. Yeah, so with this, wonderful introduction that that Tad has given about this project, you can really see the depth of the community engagement aspects of this project. So when we published our paper in PNAS, we talk a little bit about the community engagement, but we always think that it's really important to kind of try to tell this whole story, because as well as doing the science and the ancient DNA, which we're going to talk about in a couple of minutes, this we have just done a lot of work, a lot of conversations with people on the ground about what this work means to them. And I think that this is one of the stronger parts of this project, and what we hope that other researchers will, can use this as a model or an example for how they can incorporate community perspectives in their own research. So if we move to slide 18, so emblematic of that is we incorporated near the time of the reburial of the remains. So before we took DNA samples, we transported them to University of Tennessee at Knoxville. The idea was always to rebury these ancestors. And when we got closer to the date of reburial on May 4th, 2019, we held a number of ceremonies to honor these individuals and to continue to do acts that restore personhood. So the first one is a renaming ceremony. When our education and outreach director, Lachey Oubre, was doing educational outreach programs in a local North Charleston high school, one of the students asked her, do they have any names? And, we really thought a lot about that. And it is really important. We don't know the names of these individuals. They were recovered with burial numbers, so we were referring to them as CHS01 or 2 or 05. We felt that it was very important to make sure that they were returned to the ground with names and not just burial numbers. So we held a naming ceremony at McLeod Plantation Historical Site. Dr. Adeofunyan, shown here in blue, is a Yoruba priest, and he brought with him two colleagues, Dr. Natalie Washington-Wyke and Dr. Terrence Wyke, who are also Yoruba priests, to conduct a naming ceremony where libations were poured. The 5 flavors of life were given to Mother Earth to mimic how a mother will taste the 5 flavors of life for a child. The Natalie Washington-Wyke and Terrence Wyke also created names that integrated the osteological genetic findings on sex and ancestry with spiritual guidance and names sourced from Lorenzo Dow Turner's Africanisms of the African dialect. So in the future, when we're talking about these ancestors, we are going to be using names, but just know that these are honorific names. So if we go to the next slide, so on May 4th, 2019, these individuals were reinterred. And this was a huge community event. I think we had about 500 people in attendance. It was really wonderful. The ancestors were wrapped in indigo shrouds. You can see here in the horse-drawn hearse shown in black. And these are 6 of the ancestors, 2 men, 2 women, and 2 children. And you can see their caskets wrapped, and they had these beautiful indigo shrouds that were made by local artisans. They were processed to the reburial site with an Ngungun masquerade, as well with a number of cultural and spiritual leaders. You can see here on the next slide, again, that horse-drawn hearse right down King Street. Again, we had about 500 estimated people in attendance, people from the College of Charleston, from local communities, The Charleston mayor came to the site as well as city council members. So in the second photograph, you can see the big building in the back is the Charleston Gilliard Center, which is the auditorium. And that big oak tree that's to the left, that's where the ancestors were reburied, right next to that oak tree. So we have everybody kind of coming and surrounding around and being part of this really important moment in historic Charleston. We then carried 6 of the ancestors from the hearse, and you can see here with Adeofunian and Fielding's Funeral Homes carrying each one of these ancestors into the final burial vault, which you can show here. And before the ancestors were reburied, so you can see on slide 24, I think it's numbered, Dr. Adeofunian is placing messages to the ancestors. So we asked community members to write notes about what this meant to them, what they wanted to tell the ancestors, and these notes were also buried with their ancestral remains. We had a multi-faith spiritual ceremony, including Keepers of the Word, which is a Native American organization in the Low County, as well as Muslim faith spiritual leaders, Christian spiritual leaders, and as well as African spiritual leaders from I'm blanking on the name. Tad, do you remember what the name of the?
Yeah, that I'm drawing a blank on it also.
Yeah, so
It'll,
sorry,
it'll,
but a lot of really wonderful people were here in attendance, and
yes.
you can see on slide 26, we had the wonderful Children of Watoto Academy gave dance performances, and it was really an act of celebration with drumming and music and life, and that was really kind of what this whole narrative of this project is again restoring life, restoring personhood, and keeping the memory of these individuals alive, but then also discussing and bringing to light the important history and legacy of African descended persons in Charleston. So then this kind of brings us to the burial grounds ethical research design on slide 27. So we really, when we were setting up this project, and now you've seen a lot of the components, the engagement components, but they're really all centered around this philosophy of community-based archaeogenomics, which focuses on humanizing skeletal remains, so they're not regarded as objects of study, but rather people. Centering personhood in the study of these remains. So we're talking a lot about individual histories rather than population to again draw centering to this humanizing element. Also decolonizing knowledge production. So, even though we are talking about this now, a lot of the knowledge that was generated is generated from local community members. And making sure that they are the ones that are in charge of the telling of their own histories. Um, then also centering descendant communities as the primary stakeholders in this work. So, the questions that we had from the community were the primary driving questions, and we'll talk about that on the next slide. And then also important in that is interrogating our own positionality. So Dr. Schurr and myself come from outside of the Charleston community. We are also white researchers, so it's really important for us to be critically examining what our own associations are, and then how we can be actively inclusive towards other perspectives and make sure that it's about the community itself. So kind of this drove kind of our first initial meetings with the community were centered around understanding what their questions and priorities were. So we gathered a lot of questions that community members were interested in answering. So on slide 28, you can see some of these questions listed out. They wanted to know, were these individuals buried with care? What is their ancestry? Are they related to each other? What was their life quality? How did they live? And so really, in order to answer all of these questions, we really needed to build a multi-method framework. So we integrated ancient DNA, of course, which is where Dr. Schurr and myself come in, but also archaeology, archival work, osteology, and stable isotopes. And so really it's through the amazing collaboration by all these persons that are listed here, Dr. Eric Poplin, who is the head archaeologist on site, and Dr. Susan Abel, and Dr. Wolf Buchschen, and Dr. Chelsea Juarez, really were so committed to collaborating to do this multifaceted study that we were able to answer these questions with the depth that we were able to. And so I think, as anthropologists, there's really a strength when we're learning from so many different perspectives, so different ways of looking back into the past. So what we're going to do for this next section of this talk is kind of go through what each of these forms of evidence or these ways of knowing told us and what we learned about the Anson Street ancestors. So on slide 29, we're going to talk a little bit about the archaeological findings. So there were about 37 burials, 36 that contained human remains that Dr. Schurr mentioned. They're organized into roughly 4 equally spaced rows. You can see here's a site burial map to the right. You can see they have that kind of 4 vertical row quality. So this suggests that they were buried over time and that they knew the position of the burial before it as they were being interred. The archaeologists also recovered a number of artifacts associated with some of the burials. So for instance, a George III copper halfpenny was recovered from burial 27. This is Walela. Walela is a 5 to 6-year-old child, and 2 copper halfpennies were recovered, so 1 on each of her eye orbits. So this is an act of care during a burial rite where folks would place coins upon the eyes. As a way to mourn the dead. We also find things like glass beads. So, for instance, Omo, who was an infant at the time that she is buried, has these glass beads that would be for a hair ornament. So, again, this is an artifact that represents care and adornment by somebody who loved them. So, we really, again, we want to think about how care is being kind of translated through the archaeological record here. We also know the date range for this burial ground is approximately 1760 to 1790 AD, and we know that based, again, the copper coin is a really big date for us because it says 1773 on it. But then also we know based on the pottery shards that are found in the burials as well, kind of gives us that date range. The archival evidence also helps us kind of give an end date to the burial ground. We know that the Anson Street African burial ground, if you see on the first image here, is on the kind of northernmost, it's in the peninsula of Charleston, kind of in the middle. Here you can see by this red dot. But in historic Charleston around 1790, the Anson Street African burial ground is really on the most northernmost part of the city's boundary. So the white speck in the middle panel represents the modern day, the burial ground that was excavated. You can see it's right up against this north boundary street, and everything north of that is all farmland. So this is actually, relatively far away from the main city center. And we know that in around 1798 that the land the ancestors were buried on was subdivided and sold. So we likely think that the burials probably likely stopped around that point because then they were given as private property to different landowners. We also know that the land the ancestors were buried on, it was land of a white land-owning individual in kind of this northernmost area. We know that the population of free people of color, so individuals that are of African descent who are not under enslavement, were likely buried on the opposite part of the city in the east. So we don't know of any known burial grounds in this area. And then we know that based, again, on the ownership of the land in the area, this suggests these individuals were likely of enslaved status. However, it's really important to point out that we have no documented evidence of this burial ground at all, so we can only be inferring the status, and without exactly direct documentation, we don't know for sure.
I might add quickly here that this work on the mapping was significantly contributed by Grant Michaud and Nick Butler of our group.
Yes, thank you for mentioning that. And then going to slide 31 with the osteology, this work is done by Dr. Wolf Buchsten and Dr. Susan Abel of the Charleston County Coroner's Office. So they identified and assessed for age, osteological sex, and osteological ancestry. They found approximately 28 adults and 7 juveniles. And I know that the coloring is not fully labeled here, but the juveniles in this map are circled in orange, and the rest are uncircled for adults. And age range distribution goes from approximately 6 months to over 40 years of age. They also estimated sex for 12 individuals, with 8 males identified that are circled here in blue and 5 females identified that are circled here in red. And most of the rest of the individuals are labeled as indeterminate just due to poor osteological preservation. They just were not very well preserved, unfortunately. So when we did genomic analysis, we were able to also investigate chromosomal sex, and we found that 21 individuals were chromosomal males, and I think about 6 individuals were chromosomal females. And the rest were indeterminate because we weren't able to get data from that. So again, it does skew a little bit more to males, but we do know women and children are also buried here. Ancestral estimations were determined to be likely of African ancestry based on the cranium. And again, this is something that we followed up with the DNA results. The osteologists also looked at the dental health of these ancestors and found that most had very poor dental health. So we have a high presence of carious lesions, which are basically like cavities in the teeth. Abscess tooth, so that's when you have a really big carie and it's eaten up about half of your tooth. You also see a lot of generalized tooth loss, so a lot of teeth that have fallen out during the course of life, and then the hole where the root is has been healed over. We see things like enamel hypoplasia, which is a line and indentation in the enamel that occurs during a period of acute stress, usually in childhood or in adulthood. And we also see a lot of wear. So this is pretty emblematic of this time period. You usually don't have good access to dental care. And one thing that's not pointed out here is dental calculus was found on 14 of the ancestors. And Dr. Theodore Schurr will also talk a little bit about our ongoing work with University of Oklahoma with dental calculus research to kind of, again, get at this lived history aspect of the ancestors. And if we go to the next slide, on slide 33, we also conducted strontium isotopes on these individuals. This was conducted by Dr. Chelsea Juarez. She collected enamel samples and bone samples and then tested the strontium uptake of these. Strontium is a really great way of understanding place of childhood or birth, and then also of residence in the last 10 years of life, which is with bone. Strontium basically is a geochemical signature that gets absorbed into the bone through the groundwater that you drink or the food that you're eating, and is really reflective of that specific geological location or locale that you are living in. So we can compare for the ancestors their strontium isotopes to determine where they might have lived their early childhood or their adulthood. So you can see here in the first graph, encircled in red, we have about 6 individuals that have strontium enamel isotopes overlapping with values along the West African coast. So this indicates that because your enamel forms during your childhood, that these individuals were likely born in Africa and not born in the local Charleston or North American region, as the strontium values are a little bit lower. But when you look at the bone signatures, you can see really there's encircled in blue, there are only really 2 individuals that have higher strontium isotopic bone values, which indicates that they were recently brought over, forcibly transported from Africa, and then more recently passed away when they reached Charleston. So most of these individuals were living most of their lives, or at least 10 years of their life, if not longer, in the Americas. So this again gets us at these kind of life history questions. So slide 34, we begin to kind of move towards the ancient DNA. So in the context of understanding the strontium isotopes, the archaeology, the documentary and archival work, we situated our work with ancient DNA within that. So we were able to obtain whole genomic information for 31 individuals when 18 individuals had enough genomic information generated to do in-depth ancestry and relatedness estimations. We were able to obtain between 0.8 to 1.2x coverage. And I know if anybody is in here doing contemporary genomics, that looks super, super low, but that's actually okay for ancient DNA because we tend to get very not great coverage in ancient DNA because our DNA is very fragmented and in very low quantity. So anything really around 0.5x coverage is dictated to be pretty good. We called SNPs, or these single nucleotide polymorphisms, or these markers, against 2 different kinds of panels, the human origins panel to assess continental ancestry, and then a custom African reference panel, which is assembled from the literature to more specifically understand African ancestry. Our DNA was determined to be authentic. Our damage is characteristic of profiles for ancient DNA samples, around 4 to 15% damage, which is usually found at the ends of these DNA reads. And we also had relatively low contamination. So we're all good to go. And if anybody has any questions about how we get ancient DNA or how we characterize it, please let me know. We have to do this work in a full bunny suit. So you can see there's a picture of me in the full suit and the full getup in a specific ancient DNA lab doing this work. But we're going to kind of, move a little faster. So we're just going to hop right to the results. So slide 35 shows a diverse set of mitogenomes, so the mitochondrial DNA, as well as our Y chromosome haplogroups. So our mitochondrial DNA shows us the maternal history of an individual, right? So your mitochondrial DNA is inherited directly from the mother, and so it is able to show us this direct maternal history. What we see with the Anson Street ancestors is we have a majority of African associated haplogroups. So lots of L types, a U6A5 type. This is also of African derivation. And so this was this is what we expected to find. However, one individual, Kusa, so she's kind of highlighted here in this green arrow, had an A2 mitochondrial haplotype. This A2 haplotype is usually actually characteristic of North American populations. So this suggested that she may have indigenous North American ancestry in her autosomal DNA profile as well. So this was something we weren't exactly expecting and was kind of surprising. And we also found an H100 haplotype and CHS22. This is Lisa. And if anybody here does mitochondrial DNA work, you're going to say H is, oh my gosh, that's a European haplogroup. And so that was also a little bit of a surprise for us. And we're going to talk actually about this history of this haplogroup in a little bit more detail in a couple of slides. But this form of H100 is actually represents a haplogroup that left Africa and then actually migrated back into Eastern and Western Africa. So it is actually of African derivation. Our Y chromosome haplogroups reflect, again, a strong African ancestry, E1B1A, E2, and then a B2A. So again, this was something that we were also expecting. It's very common in Bantu-speaking populations in West Africa. On slide 36, we did find two individuals that shared the same mitochondrial DNA haplogroup, L3E2A, that was shared between EC and Wilela. Remember, Wilela was that child with the coins placed upon her eyes. So this can indicate that potentially there is a mother-child relationship, a mother-daughter relationship here. However, unfortunately, we weren't able to get enough genomic DNA for Wilela, so we couldn't verify this using autosomal ancestry, which was kind of a big bummer. And unfortunately, between the 18 individuals with higher amounts of whole genome data, we were not able to detect any autosomal relationships between them. So that means we weren't able to detect any parents and offspring or sisters and brothers between them. So what this indicates to us is that these individuals were interred likely via time. So as one person passes away, the next person is interred instead of by biological family. And so again, this kind of changes our arboreal interpretation for this space and is hard to reflect upon because we don't see these biological kinships association. However, what we do see again are those acts of care by people who did inter these individuals through the archaeological evidence. So moving into the ancestral estimations, which is kind of the main point of our PNAS paper, we first did a PCA, a principal component analysis between shown here against the human origins reference panel. And what we find here is that most of the individuals are labeled here in these light blue symbols. And so you can see them projected onto this PCA plot. Basically, a PCA plot is showing us the distribution of genetic variation between different reference populations. One dot equals one person, and each color represents one population. So you can kind of, by projecting where the ancient DNA individual is on this plot, you can kind of begin to infer autosomal ancestry estimations. So again, this largely reflected what we see in the mitochondrial and Y chromosome haplogroups, largely of African ancestry, except for we have two individuals circled here in green and in red that are projecting a little bit slightly off from the rest of the individuals. And we're going to talk a little bit about those two. So the one circled in green is Kusa. And again, remember she had an A2 mitochondrial DNA haplotype, which is of Native American, North American ancestry. And then Lisa, which is circled here in red, I remember she had that H100 haplotype that was a little bit unusual. So next we did an admixture analysis. The shown here is that K equals 11. Admixture basically allows us to look at population clusters. So we have our big reference panel and a specific number of population clusters that you tell the program. It will calculate based for each individual what ancestral profile they have. So just remember each line represents one individual, and then we've grouped them by continental, so you can kind of see European populations tend to have this big royal blue component versus African populations tend to have this nice light blue component. So what we did, and we compared that data against the ancestry ancestors, we found that majority of them again had this nice light blue, which indicates African ancestry, which makes sense. However, one individual, Kusa, also has this light brown population cluster component. And when we look about where else is this light brown population cluster, we can see here it's found in the Americas. So this reaffirms what we found with the mitochondrial DNA, which is a slight part of her ancestral background is of Native American ancestry. So next we ran a PCA plot. This is on slide 39 with just African population. So here we are just showing West and West Central African ancestry. We have a bunch of other plots that for brevity's sake, we're not going to answer. We're not going to show, but what we do see is that a wide distribution across the first PC axis, and we have basically half of the ancestry ancestors projecting with West Central African populations and half with West Central, with just Upper Gold Coast populations. So this indicates that these individuals have diverse African ancestries from across the western coast of Africa and not from just one centralized location. Now, specifically, if you see it highlighted in green here, or highlighted in blue, circled in blue, one person is projecting a little bit farther down with populations in the Gambia. So this individual is Lisa, and we're going to talk through an admixture plot. So she had a very distinctive PCR projection and also had a very distinctive admixture plot. So we ran, again, here's the Lisa's has a very lime green profile. And gosh, I'm sorry because I didn't realize that I had animations on this one, so you can't see. But if you look at that first population, Gambia, and you look all the way to the left, there's going to be a very light green ancestral profile. And if we zoomed up on just Gambian populations, you would see that ancestral profile fits with Fulani populations, which is really exciting. They have a very distinct genetic admixture component. And then on slide 41, we actually see that our mitochondrial DNA actually confirms this connection to the Fulani. So her H100 haplogroup is also classified as an H1C1B1A haplogroup in the literature. And when we do a reduced meaning and joining network against this compared to other samples from the Gambia, from Burkina Faso, we see that her haplogroup most closely associates with those found in Fulani individuals from Niger, Guinea, and Burkina Faso. So again, we see from multiple different lines of evidence her connection. So now I'm going to turn this over to Tad. I'm sorry if I talked a little long.
Okay, well, thank you, Raquel, Dr. Fleskes. I'm going to turn attention now to some ongoing work and some future directions. Although I also and talk a little bit about at least touch on a couple of points about the DNA work that Raquel has mentioned. So she also had indicated that in addition to doing a basic osteological examen of the dentition of the ancestors, we were also able to successfully retrieve dental calculus or essentially ossified plaque from the teeth of 14 individuals and then use them to extract DNA from the calculus to look at bacterial compositions of the microbiome of the mouth, the oral microbiome. And so what you can see in the slide following that, I guess, what is that, 41? And I can't tell what number that is. But anyway, you can see that part of what gets trapped in the mouth are both food particles of various kinds, proteins and byproducts of the foods, but also the oral bacteria and potentially viruses or other microorganisms, which then are reflective of the oral environment for the individual whose dental plaque that we're analyzing. And we're most interested, as you can see in the next slide, just in a very simple cartoon of the bacterial flora, the various species of bacteria, which tend to be linked to the kind of foods that we eat and the relative number of oral pathogens that we might have in our mouth because of cavities or periodontal disease or other things like that. The slide following that shows, at least in a schematic way, a whole variety of things that may be indicated by studying the oral microbiome of living persons and presumably also, though, of past populations. Certain kinds of taxa or species that may be a higher or lower for cancers or ulcers. Some may be higher for, in the upper right quadrant, you can see that periodontal disease may be linked to Provotella, and on the far, oral cancers on the far left, and possibly other things related to more complex diseases with regard to other species. So we are interested in knowing what kinds of bacteria were in the mouths, literally, of the ancestors at the time of their death, and what that might say about the food they ate, their dietary health broadly, oral health, and maybe any kind of oral diseases they might be predisposed to or having, or the risk factors that they gave to them that might have something to do with their cause of death. The next slide shows you the laboratory and the group we worked with to carry out this work at the University of Oklahoma, which is specialized in ancient DNA work with microbiome and in dental calculus of a variety of species and or populations, and also different human populations today. The team that the photograph shows all those persons who are involved in this component of the work and the collaborative team that we're using for the studying with the dental calculus analysis. The next slides show you again people in bunny suits, including Dr. Fleskes and I, and also Sarah Johnson, who's a doctoral student there working with us on this project. And the next two slides show Raquel actually doing the processing and beginning work. Actually, maybe was it library preps, or I think at that stage with the DNA, beginning to prepare the DNA for actual sequence analysis. So the dental calculus results are the next series of slides. As it shows there, that we were able to successfully retrieve dental calculus of 14 ancestors, and then successfully extract DNA for analysis from 12 of those 14 persons. Once we did the whole genomic shotgun sequencing for the metagenome or the bacterial sequences, we were able to successfully do so, at least in ways we think of allow us to do further analysis of 11 of those 12 persons. The source tractor profile in the next slide shows you pie charts. And what this essentially is showing you is, because the metagenome or the DNA that you sequenced, it may be bacterial in terms of its overall makeup, but not all of it is necessarily coming from the oral microbiome. Some may be coming from soil bacteria, other sources because we're taking them from persons who were interred and buried, and they may actually have accumulated those kinds of bacterial types in the process of diagenesis, and also and also being disinterred from the graves where we found them. So the purple here is the important part of it, because it shows the proportion of bacterial types that we think are going to give us, are actually probably authentic to the ancestors themselves, and will allow us to go forward with various sort of sequence analyses. In fact, that is the case, because you can see on the next slide, the oral bacterium pathogen slide, that this is a heat map showing you some number of ancestors, I think along the bottom indicated, and then the number of bacterial species shown on the vertical axis. And the heat map simply indicates the relative types of, or taxa, or number of species of bacteria, or genera of bacteria that are present in the mouths of the ancestors. That we think are going to be important for telling the story about oral history and possibly periodontitis and periodontitis and things of this kind. The next slide shows you how many species we've mapped, and using Metaflan and other techniques to try to characterize them at a more of a species level. The main point here is that these seem to be highly enough represented and having specific sort of metabolic functions, that we're going to focus our attention on the phylogenetic analysis of these species to figure out what they might do, what they look like in the ancestors, and then compare those sequences to those coming from living persons to see how bacterial species belonging to these taxa actually have evolved over several centuries of time. But we believe these are going to be key species for telling us something about, again, periodontal disease, oral health, things of this sort, that will be maybe even give us clues as, again, to the lifeways, and possibly maybe some broad insights into cause of death. In any case, And if you want to add something here, Raquel, you can. Or you.
No, I think you did a really great job.
Okay, excellent. So what we're doing then, just as an overview, again, what we're doing is identifying dental species and looking for novel types in the role in health and disease, and thinking about how this relates to periodontal disease. So they're important because this is actually some of the first dental calculus study to be done for individual archaeological populations from the colonial era in North America, and certainly that for African descended populations. And furthermore, this may actually tell us something about the generalizable maybe trends for colonial African populations or enslaved African persons, just based on the diets they had, or possibly differences between those individuals living in more urban versus rural contexts. But this is all at this point somewhat speculative, but we have hope to be able to do something more along those lines. And again, the basic questions that these data will outline, these key questions, you can see how they're all here. And these are also questions in some general sense that the communities we were working with actually wanted to know more about. How did they live? What did they eat? What was their life like? And how did they die? And in various ways, we hope that both the DNA evidence, but especially the dental calculus data, will tell us those kinds of things. So the work in progress, and I think here also Raquel will add a few points. Where again we published work on genomic evidence data from the ancestors, and now the next steps are to actually compare those data to the data from living Charlestonians, African Americans who are participating in this project, and actually more broadly with Gullah Geechee communities across the Low Country of South Carolina. And also, we're doing extensive education and outreach efforts to help bring some of this information to public schools, with public lectures and things of this kind, and more broadly with these publications and with our ongoing community presentations in Charleston to raise awareness of this African burial grounds in the Charleston area and the importance of acknowledging the existence of these folks, their importance to the history of Charleston, and recognizing them as persons who otherwise are basically unknown and unacknowledged in history. So, would you like to add anything beyond that, Raquel?
Yeah, I think Tad has summarized this really well. I think this work has a platform right now, and maybe Tad, you can talk about this a little bit too. But we're more moving towards creating a permanent memorial on the burial site. So remember, if we were talking about the reinterment ceremony and where all those ancestors were reburied, and we had consultations with community members, the events leading up to their burial. Well, the Charleston City has now sponsored a memorial with Stephen Hawes, who is an artist from North Carolina, who has done a number of really amazing work about the African diaspora. So they had events, I think it was last weekend or 2 weekends
Yes.
ago, where they
Yep.
cast hands of 36 community members representing each of the 36 individuals. These hands will then form around the diameter of a fountain, and create a larger area of place for reflection, and to bring community members in to do educational events to learn about the ancestors. So this work is kind of an extension of how we do science with again with community impact, and with the effects of this work expand much outside of the scientific realm.
And again, just to emphasize the point that this all is being driven by community interest and their own, us also bringing forward their stories about their families, not just those of the ancestors, to allow us to see have their take on this history, which in various ways is untold, or at least not formally told, and for us also then to give visibility to them as important members of the community as well. And speaking of that, unless the Raquel would like to add something else, I'll pause for that. But I wanted to make sure we acknowledge Dr. Adia Funyan, who sadly left us to join the ancestors a couple of actually now 2.5 years ago, and who was a central figure in getting the Gullah Society off the ground and all this work we described started, and whose memory we honor with everything that we do in terms of talking in presentations like this or publishing our work and so forth. His spirit lives on with us in a significant way, and we're enormously grateful for his involving us in this project in the first place. Would you like to add anything to that general sentiment?
I think maybe I'll just close out with some acknowledgments.
Okay, fine.
Yeah, this work would not really have been able to be possible without our collaborators that are listed on this last screen. So, Coastal Community Foundation, University of Tennessee, National Geographic, that we were able to obtain funding through this, as well as the College of Charleston and Gullah Society. And now our team is under the Ancestry African Burial Ground Project. So we'll link our website. It's asabg project.com. And if you want to kind of keep following our work and the ever unfolding impacts of this research. And thank you very much for having us
Yes,
here today.
thank you
We
very much.
really appreciate being able to talk with you all.
Well, thank you so much for presenting this wonderful work. And I already love the paper because of all these different aspects that kind of guide your research. But after listening to your presentation, I have to applaud you even more, and I see comments in the chat about the excellent work you do. Dr. Olu, who comes here all the time, and he appreciates the strong scientific rigor integrated in important cultural sensitivity. And Dr. O says, excellent work. I really appreciate the relationship you have with the community, and so on. So it's really, I really am also really happy to hear that that the renaming took place and that you first went into the communities and asked what questions you want to have answered. I think we should include that in research in general more. Um, and it's far too rare, even in medical research or clinical trials. We imagine what people's problems are that we don't have personally, or what answers or problems people want solved in different situations that we are not in. And most of the time we spend our money and time on things that are really not close to people's problems and hearts. So I wanted to really emphasize that that you're doing that, and that's wonderful. And I hope in moving forward a lot of more research groups do that. So yeah, thank you for that first of all.
Thank you.
Thank you.
And I also really appreciate that you go into the schools and that education in those communities um, are participating. Is also the scientific part being discussed, like how DNA, like how it gets inherited and how you extract it. Is there something like that also going on? That would be really wonderful, but I know it's probably really hard to have that in a school. I don't know.
No, no, yeah, you're hitting on some really great points, and unfortunately, we can't cover everything. But yeah, the education arm of this project was a very essential part of this work. So when this project was getting started, Dr. Shore and myself traveled down to Charleston almost every 3 or 4 months, and a lot of that time was spent going to local schools, elementary, middle school, high school, college level, to talk about this work. And a lot of that is public science outreach, right? How, when we're talking about ancient DNA, what is that? It can be kind of complicated, but we really wanted to make sure that folks really fully understood how these processes were being carried out, what was happening to these ancestral individuals in order to get the genomic data. And then it became a project around, public science education and outreach and increasing visibility and transparency in science. We brought a GoPro into the lab and filmed the process of doing ancient DNA in real time, so that when we were next time back with the community, we could actually show them how the process was being carried out. And maybe Ted, if you want to talk a little bit more about this.
Well, I would just amplify those few points, which I think are really important takeaway from what we're doing as a means of actually giving opportunities for kids who normally don't get to learn about STEM related research or archaeology or anthropology in their classrooms, or even much about Black history to some extent, in the way, at least the way that we're approaching it from this project. And so our goal, is in some broader sense to build capacity or to inspire young people to do this work, to feel like they can do it, to see what it's all about, and to attract their attention to new kinds of science that might be engage them going forward, as well as actually, I think, educating the teachers who are also teaching them about science and biology. So it is a very important part of what we try to do on a regular basis as part of this research project or this broader project.
Yeah, I think that's so important and interesting. And by coincidence, I was talking yesterday with people from that invested, for example, here in Clubhouse and other things. That we were talking about science, society, and science education or communication, and it was really interesting. This lady from Iran said we should bring the science close to people's heart and their everyday life relevance, and that would basically bring people closer and have a different relationship with science, and it's not something scary, and you're doing exactly that. So um, I think that's really important and really interesting, and I hope people will be scared less of science and see it an enrichment of their personal world also through that. So I think it's not just a contribution to the field, and people in Charleston is in general that approach is an enrichment of society with science, the relationship, I think.
Yeah, I think, there's a lot of trust building that science has to do, given a lot of inherited histories of the past of science maybe not looking out for folks' best interests. And so we really view it as our responsibility to make sure that folks really they trust us, they know what's going to happen to these ancestral individuals, that we're not going to be using the data for purposes other than what they have specifically prescribed. And in fact, all of the DNA samples and all the remaining bone material was reburied at the time of reinternment. So we're viewing any part of these ancestral individuals as being, important and to as a part of that one individual, and also deserves to be put back into the ground. So there's no disconnect.
Yeah, and I wanted to read out one more question, and then I wanted to give Joyce that came up here to the stage an opportunity to speak. I know the hour is up. The one question by Alex Solan was, where can we find similar studies, or is um, in other parts of America, like Maryland and other states, is something similar going on, or are you planning to do something similar in other communities?
Um, well, right now there's, I mean, I think our project is the largest. I know I'm doing work right now in the Maryland, Virginia area. We're doing a great project with the First African Baptist Church in Williamsburg, Virginia, which right now we're working on 3 ancestral individuals that are about 1820s. And then I have another site I'm working on in Delaware, which is a 1680s site, and 3 African descendant individuals are being found there. But this is really very new work. And so, and it does take a lot of time because you have to be able to form all these community connections in order to really do this work ethically, with a strong ethical foundation. So I hope that in the future that, this work can expand. I mean, it's really exciting, uh, and offers, again, another view into history.
Yeah, thank you. And Joyce, did you have a question? Welcome to the stage.
Yeah, thank you. Well, I wanted to also agree with Katarina. This is really great. It's so respectfully done and integrating the cultural aspects. That's really wonderful. And also, I wanted to also agree with Katarina about including participants' perspectives, which I think is important in medical research as well. But also, with regard to the dental work, I know that I read a study a while back where they were noting that when people started to, even before they were fully sedentary, when they were just part of the year staying in area and like grinding up acorns or something, that's when they started to have the dental caries, because they were changing the texture of the food and it was changing the microbes and so I was wondering, are you comparing? I guess you maybe haven't done it quite yet, but is the dental caries situation worse in these samples than in Africa, where they may have come up with ways of keeping it in check by using like native plants or something to clean their teeth or things like that? Anyway, thanks.
That's a great question, and thank you so much for your comments. So we have not yet. So I'm not an expert in dental pathology by any means, but I do know that the ancestors did have pretty poor dental health, and I think likely that's due to the types of foods that are being eaten. So things like rice and corn and all that. It's very starchy and sugary, and that translates to a lot of buildup of dental plaque that can happen. So I don't know relatively how much that differs between other populations, let's say on the African continent or in different time periods. But I do know that, especially in the colonial period, you do have a rise of very, very bad teeth just because you're eating a lot more of these starchy substances, and that can really erode the teeth very badly. So what we're trying to do is also compare the specific, if we have high enough coverage for genome reconstruction of certain one of these pathogens, is to compare them with other pathogens that are found in historic dental calculus samples or in contemporary dental calculus samples to see kind of the evolution of these specific microbes in the context of things like the agricultural transition, or is there any kind of continental associations of where this microbe itself developed. So these little individual microbial histories in the context of kind of, again, these kind of the buildup of the dental plaque itself are something that we're really interested in pursuing.
Yes, thank you.
Hi, LT, and did you have a question?
Yes, excellent talk. I really enjoyed it from beginning to the end. My question has to do that you mentioned that when you're doing the sequencing, because it's the ancient DNA, it's ancient, like old. So you the clean lab. So my question has to do with is it like how commonly you set up let me change the question. It's been a long time since I've been in a lab. So, meaning that, yes, meaning that, do you have to do a special, like a requirement, like setting up and you have your own clean lab, or you go to some others university, other sites clean lab? How many such clean lab is in the US? I guess to be more specific. Thank you.
Thank you so much for your question. I love that you asked that, because I felt when we were giving this talk, I was like, oh, we're skipping over all the cool stuff, which is talking about these clean lab setups. So there are probably about a dozen in the United States that have this type of setup. It's kind of based on like a BSL-3 type of lab, except we're really trying, we're not working with any pathogens here. We're just really trying to control the DNA from the researchers, not getting into the DNA sample of the ancestor. So we have to work in usually multi-chambered labs that are ISO 6 or 7, that have overhead UV lights for cross-linking any DNA that's present or found that's not in the sample itself, as well as positive pressured HEPA airflow. We have to wear full PPE, gloves, masks, Tyvek suit, boot covers, shoe covers. Our hands must be routinely washed with 10% bleach and 70% ethanol. Everything has to get fully cleaned after every sample. So a lot of this is just a lot of labor and time to make sure that we're really, again, not mixing up the DNA between samples, but then also making sure that we're not getting the DNA of the researchers themselves into the DNA sample, because the DNA from these ancestral individuals is so highly fragmented and in such low quantities. What can happen is that if you have contamination, it can really just eat up all of the sequencing signal, and really take away from really a highly endogenous or a DNA sample itself. So it's quite a robust and laborious process, but the end results I think are worth it.
Okay, great. Thank you. May I ask another question, Katerina? Do you have time? Sorry.
Yeah, as long as Raquel and Theodore have time, please go ahead.
Okay. Okay. So my next question is, you mentioned a little bit about there's no autosomal relatedness. So you give some kind of explanation. Can you just comment a little bit more, because it's a long talk. I kept thinking back. I did have. Can you please just a little
Of
bit?
course.
Thank
Of
you.
course. Yeah. So when we're talking about autosomal relatedness, we're basically comparing the 23 or 20 genomic reads from our 23 chromosomes to determine, is this a parent offspring? We're doing this using what is as a pairwise estimate of relatedness, as what through the read. This is a read program, as well as kind of we do R1, R2, or R0 and King robust estimates of relatedness as well. And so but basically, unfortunately, because we only had 18 individuals with high enough genomic coverage to make these estimations to be robust, we're only able to limit our estimations between those 18 individuals. And so when we're looking at mitochondrial DNA, mitochondrial DNA are much, much smaller. It's only 16,000 base pairs. So it's much easier to get good coverage with the mitochondrial DNA to be able to hypothesize about this maternal relatedness. But what we really would have loved to just be able to verify it, because either you can have the same mitochondrial DNA haplotype and be a parent offspring, or you could have that haplotype by chance and really share a much deeper ancestral connection. So if you have an L1 haplotype and the other person has an L1 haplotype, you could be direct parent offspring, or you could share that relationship, 10, 13 generations back. So we were really hoping to get that autosomal coverage for Willela, but unfortunately, we just didn't have enough to be able to verify. Does that answer your question at all?
Yes, yes. I missed the part because you said the only half of the sample you got some kind of good coverage. Thank you. That's the part I was wondering. Okay, thank you. Yes, no more questions.
And I might add that from the Y chromosome standpoint, what Raquel characterized as one of the interpretive challenges regarding direct ancestry or more distant relationships holds true for the Y chromosome, which is actually substantially larger than the mitochondrial genome. And with the lower coverage of the genomic sequencing, we don't necessarily have the same regions to compare for all the individuals who are male in the population. So we still can find haplogroup, Y chromosome haplogroup associations, but we have to be careful about how we interpret them and try to, as Raquel says, use autosomal data or other means of inferring relatedness to make sure we're making the right interpretation.
Yes, thank you so much. I got that too. Thank you.
I don't know if you have time for one more question. Noel joined the stage, and I think that would be the last question, if that's okay with you.
Sure.
Yeah, that's fine.
Hi, Noel.
Hi. Oh, hi. Thank you so much for your work. I'm from the South, so I find this really interesting. Um, I was wondering with this grave site, there's a lot of questions I had, but I'll just stick to one. With this grave site, and I know there was like so much migration that happened during that time frame between the Seminole and Gullah Wars. Do you think that you could be able to tie that kind of activity to these particular people, um, or is it just kind of like people that you found that are in that location? I don't know if that makes some sort of clarity about what I'm asking, but do you think they were attached to it or were they local?
I think, Raquel, you can add something to this. I'll speak to the Gullah connection and to respond in part to your question. We've done work with Gullah communities besides those in South Carolina, living persons whose genomic profiles we can compare to those of the ancestors, and that may talk about the relative degree of connection between the ancestors and living members of the Gullah Geechee community, or at least in general genetic profiles. So that may give us some clue as to how coextensive that the ancestors were with the broader Gullah Geechee community or the community that was forming at that time. How they're related to that, the conflict that you're talking about, or that interaction, I don't know that that's clear, but it's the work showing Kusa having Amerindian ancestry, both on the maternal line and her autosomes, suggests that there were interactions between those communities who were often, the native persons were the first to be enslaved in Charleston area, and then people of African descent. So being of somewhat lower social status and also being in the proximity may have led them to form partnerships or social relationships that were are reflected in the genetic makeup of African Americans today, actually among Gullah Geechee, but also those living at that time among the group that is represented by the ancestors. So I think we can say things of that kind, but how well we can pin or associate our data with specific historical events, I'm not sure we can do that as of yet.
Yeah, and to kind of piggyback off what Tad is saying, I think that's a really important historical point that you bring up, Noel. Unfortunately, because we just don't have good documentation about who these individuals were in life, it's really difficult to tie them to any specific event. What we're having to do here is really just use all, what we can from archaeology, from the DNA, from what we can kind of piece together with maps about to try to infer about who these individuals were. But it's really hard when we don't have anything that directly ties them, because really that's the gold there. That's the bread and butter is, being able to actually find direct documentation that could tie these individuals to a specific cultural group with specific history or to that period of time that you're referring to.
Thank you.
Thank you.
Yeah, great. Thank you so much for coming and presenting this really wonderful work. For many reasons, it's wonderful work, and also to take the time to answer our questions. Um, it was a really wonderful discussion, and again, I want to we are honored having you here, and then we want to congratulate you for this wonderful work, and we will be following your work. I read in the chat that people are curious to learn more about the different, ongoing research. So I'm really glad that you came. We got to meet you, and yeah, that we will have now more people interested in following your work. So thank you so much, and we wish you all the best for the future.
Thank
Thank
you
you
for
very much,
having
Katarina.
us.
Yeah, and thank you everyone for coming, asking questions. It makes these events so much more interesting. And if you like discussions like this, follow the club. The next room will be next week, and then we will talk about climate change and how heat wave change patterns in the soil moisture and how these patterns exactly work in different climate models. So thank you again, Raquel and Theodore, and everyone, and I hope to hear you all back soon one day. Thank you so much. And close the room in 3, 2, 1. Bye, everyone. Thank you.